Alzheimer's disease is a neurodegenerative disease known to be prevalent in a significant proportion of the elderly population, with symptoms most commonly being displayed in an individual's 70s. It's the most common form of senile dementia, with multiple hypotheses to the cause. Patients experience varied symptoms as the disease affects individuals differently. The symptoms can be classified into two main groups, both cognitive symptoms and physiological symptoms. Patients can experience cognitive symptoms starting with the loss of declarative memory, followed by psychosis, hallucinations, and delusions. Psychological symptoms include brain atrophy, especially in the cerebral region, as well as narrowed gyri, widened sulci, and enlarged ventricles. Post-mortem materials also show the presence of extracellular plaques called peptide, amyloid peptide, surrounded by swollen axons and dendrites. As previously mentioned, there are physiological symptoms present in the central nervous system. The most obvious is the reduced brain size, as can be shown here in MRI scans. Difficulties arise in animal models due to the uncertainty surrounding the cause of Alzheimer's disease. There are three main hypotheses to the cause: the cholinergic hypothesis, the amyloid hypothesis, and the tau hypothesis. The cholinergic hypothesis states that the primary cause of Alzheimer's is the degeneration of cholinergic neurons in the basal forebrain and thus the loss of cholinergic neurotransmission in other areas of the brain. The amyloid hypothesis arose due to the post-mortem examinations finding the presence of amyloid plaques. The deposition of these plaques occurs to the processing of the amyloid precursor protein. The surrounding areas around the plaques become swollen, disrupting the normal neuronal function. Used in conjunction with the previous amyloid hypotheses, the tau hypothesis states that the accumulation of the abnormal tau protein forms bunched neurofibrillary tangles that disrupt the individual's cognitive function. The most common cause of Alzheimer's is probably a combination of the three hypotheses. As previously mentioned, Alzheimer's disease is a multifactorial disease, so it's possible that we got the program to have the disorder in our genes. Genome-wide association studies have identified possible risk genes associated with Alzheimer's. These genes include the APOE, amongst others. Knowing these genes allows us to produce transgenic animal models to investigate. As you can see here are some of the risk genes identified through the genome-wide association studies. We can see that APOE and BIN1 genes are prevalent in people diagnosed with Alzheimer's disease.
So fruit flies have been used as a model of Alzheimer's disease because they are cheap, it's fast to raise them, and it takes 10 days to progress from an embryo to an adult fly. And 70% of the human disease-causing genes actually have an ortholog in fruit flies, so it makes it really easy for us to do genetic studies and analyze that. Surprisingly, fruit flies have a similar brain organization to us. They have a tripartite organization, so they have the proto, deuter, and tritocerebrum, which is similar to our fore, mid, and hindbrain. Because of fruit fly studies, the main benefit of them is the eye morphology, and basically, if you change the phenotype of the fruit fly, you can see the effects through the eye changes, so the eye color becomes a different color, and depending on that you can screen the fruit flies faster and see the effects faster just from looking at them. Also, that the neurotoxicity studies have been prevalent in fruit flies, as buildups are quite common in them. But there's no ethical issues of animal testing when it comes to fruit flies, so the benefit is that you can test first in vivo models of a lot of dangerous drugs can be done on fruit flies without worrying about putting them onto mice or even primates. So the current fruit fly Alzheimer's models are based on overexpression to see the disease and how it progresses, or silencing it to see how these proteins would function in normal pathways. So an actual ortholog gene exists for the amyloid precursor protein, as well as the beta and gamma secretase enzymes. So we can engineer them to see how they progress faster. There's also neurotoxicity studies done on how the AB protein causes the plaques and degeneration of the brain. It's faster because the fruit flies only have an 120-day lifespan. It's faster to see the progression of aging and how fast it affects their memory and cognitive function when they have these gene engineering studies done on them. So they also have, like as we mentioned, there was the tau hypothesis and the APP hypothesis, and the awful log of tau exists as well. When they did studies on tau models, they showed that there was a degeneration of the cytoskeleton, of the axons, and a change in the actual morphology of the neuromuscular junctions. So fruit flies are beneficial because they can allow us to study the neurogenetic process on a much faster scale, and they're a rapid immune system. Fruit flies have been used to study natural remedies for Alzheimer's, such as ginkgo biloba, curcumin, and Chinese herbal medicine. And a recent study on biopharmaceuticals has found that a genetically engineered protein Affibody was able to, when administered to fruit fly models of Alzheimer's disease, no symptoms occurred even with overexpression of genes, and it actually reversed the formation of the protein aggregates and it promoted their clearance. So Affibody could be a first-line treatment for Alzheimer's in the future, and it has only been tested on fruit flies thus far.
So the animal models must accurately represent the pathology of Alzheimer's disease in the early stages, as treatment is more effective in the early stages, so the preclinical stage rather than after the onset of disease, which is more difficult to treat. One model representing the amyloid beta protein pathology is transgenic mice, which overexpress the amyloid precursor protein, APP. These mice have an accumulation of amyloid, which is a key feature of the amyloid pathology. However, other amyloid precursor proteins are overproduced, which are not relevant for Alzheimer's disease. This therefore limits the phase validity of these models. To overcome this limitation and improve the phase validity, mutations are introduced into knock-in mice, which cause overexpression of only amyloid beta, and specifically the amyloid beta protein 42, which without overexpressing other amyloid precursor proteins. So these models were generated by humanizing the amyloid beta protein sequence by modifying 3 amino acids that differ between humans and mice and introducing the following mutations. The Swedish mutation KM67-671NL and the Berutha-Iberian mutation 1716F. These mutations increase levels of amyloid beta protein 40 and amyloid beta protein 42, and also the ratio of these two proteins respectively. These mouse models are able to model the neuropathological features such as cognitive impairments, build-up of plaque, and neuroinflammation. However, the limitations are that it's not representative for neuronal cell death and neurofibrillary tangles presented in Alzheimer's disease.
Okay, so another type of animal model gets used is the non-human primates, which is split into what gets called the Old World monkeys, New World monkeys, and the Great Apes. There's around 65,000 of these are used in experiments every year in the US, and another 7,000 across the EU. Their use is quite controversial because although the high level of gene conservation between these primates and humans means that the brains are very similar, that comes with an ethical challenge because there's sort of increased likelihood that they'll suffer like pain in the same way that humans will. And yeah, some of the most publicized attacks on animal research facilities have been because of primate use. And a lot of primate researchers in the 80s completely abandoned all their studies as a result of this. It's macaques are the most commonly used, which account for around 80% of the UK tests and about 65% in the USA. Some advances have been made using NHP research, include a polio vaccine, Parkinson's deep brain stimulation, and HIV drugs. The complex social behaviour and cognitive capacity of non-human primates raises questions about the ethics of their place in animal research. Some believe that the sentience displayed by many of these animals gives them moral standing, a concept that would equate their rights to that of a human being. While non-human primates exhibit aspects of rationality and agency, they do not reach the level seen in humans, and their sentience is the main factor that is said to give them the moral standing. The Great Ape Project argues that the great apes, which encompasses gorillas, orangutans, chimpanzees, and bonobos, should be given the right to live, protection of individual liberty, and the prohibition of torture as a result. Although, as of 2015, Spain is the only country that has put this theory into law. What time now? Non-human primates serve as an important translational model of age-associated amyloid beta deposition due to their phylogenetic and physiological similarities to humans. A 2019 study found increased levels of amyloid beta 40 and 42 in elderly Caribbean vervets compared to their middle-aged counterparts. Higher levels of tau were also detected in the temporal cortices of the older animals. So the study concluded that the non-human primates displayed human-like amyloid neuropathologic change. However, the results from tau did not mirror the human studies in the same way. So the researchers emphasised the need for more studies involving models that more accurately represent the complexity of this disease, such as non-human primates.
Zebrafish can also be used as animal models of Alzheimer's disease. The zebrafish are small, hardy freshwater fish native to India and often kept in home aquaria. They were originally used as a model organism for the study of vertebrate development. Zebrafish embryos can often present a vertebrate system in which to examine the molecular and cellular functions of genes implicated in Alzheimer's disease. The zebrafish genome is extensively annotated. The zebrafish evolutionary lineage separated from the human lineage approximately 450 million years ago. The zebrafish possess genes orthologous to the human genes that are thought to play essential roles in Alzheimer's disease. There are various characteristics that make zebrafish a versatile animal model and make them advantages over mammalian models. For instance, they lack the advanced cognitive behaviors as evident in rodent models. This makes them more simple and easier to study. They have transparent embryos. They rapidly develop outside the uterus. They have a large reproductive capacity. They have multiple genes which can be manipulated simply and effectively in the zebrafish at physiologically relevant levels. There are particular manipulability due to their large size, their ready availability, the ability to exploit changes in their development for assay of particular gene activities, and also owing to the simplicity of their natural habitat, it is much easier to maintain them in a laboratory setting than to stimulate the conditions essential for animals. Thus, zebrafish can be grown in a cost-effective manner. Zebrafish are also an advantageous model for genetic and molecular studies. Zebrafish embryos can be genetically malleable by injecting morpholino antisense oligonucleotides, mRNA, transgenes, and more recently by genome engineering systems. These technologies can make both subtle and drastic changes in gene expression with the effects observed in the developing transparent embryos. For instance, morpholinos are designed to bind to particular sites and transcripts from a gene of interest. Binding of a morpholino can either block mRNA translation, which is known as a knockdown effect, or interfere with correct splicing of exons. Also, injection of sense mRNA can allow overexpression of a particular gene of interest. Zebrafish possess genes orthologous to the human genes that are thought to play essential roles in Alzheimer's disease. For instance, an example on these genes are the PSEN1 and PSEN2 genes that are orthologs of the human PSEN1 and PSEN2, respectively. while the APPA and APPB genes are also orthologs of the human APP gene. There are neuroanatomical similarities and behavioral similarities between zebrafish and humans. For example, research shows a high conservation between zebrafish and human brain organization. Similar to mammalian brain, zebrafish brains harbors the main excitatory glutamate and inhibitory GABA neurotransmitter circuits along the presence of muscarinic cholinergic receptors. As for behavioral similarities, there are several anthropomorphic assays that have been performed to depict similar behavioral pattern between zebrafish and humans, which implicates a conserved behavioral mechanism and circuitry paradigm of both systems. For instance, one study has shown the cholinergic neurotransmitter system modulates drug-induced reward activity in the zebrafish, making it compatible to study the neurobiology of addiction. Finally, zebrafish can be studied and can be used as a neurogenetic model. Studies have shown that APP knockdown zebrafish displayed defective convergent extension movements with reduced body length and short curled tail. Another group has showed that the mild type PSEN1 gene in the zebrafish facilitates amyloid beta 1 to 42 secretion, similar to mutations associated with Alzheimer's disease in humans. It has also been found that loss of PSEN2 gene expression hinders the synthesis of dorsal longitudinal ascending interneurons from the spinal cord during the development of zebrafish. In conclusion, the zebrafish therefore harbor a huge resource of genetic information which need to be manipulated to reveal the molecular details of Alzheimer's disease. Okay.
Oh, we've got to leave it on that for a bit as well.